Uh, Good morning. Sorry for the delay. Good morning, Abhiniti. Good morning, Nivedita. Good morning. Jeena Priya, good morning. Akshaya, Shanti P. Good morning. So today we have one more lesson to cover in botany, right? So that is environmental issues. So first we will go through the geology aspects and then we will go to the botany aspects. Still many members has to join. Raksha, good morning. Okay, so an important announcement yesterday also I told, that is there will be two types of videos. One is outline session and we'll be having full chapter part wise, right? Within the chapter subtopic wise we'll be having. So that is one playlist. So that is going to start from today. So every day from 2 to 3 one live session will go, and at the same time 7 to 8 one more live will be gone. So 7 to 8 will be full chapter biology topics will be taken up. Biology chapters will be taken up, both 11th and 12th. And 2 to 3 would be outline sessions. Now what we are having is a quick go through NCERT highlights and important topics, right? But 2 to 3, right? So from today onwards, the 2 to 3 p.m. Live sessions would be, you know, I will be taking one chapter and I will be going in depth, briefing almost all the topics, and within 1 hour I will try to cover the lesson. Okay, so it will be a it will be again a revision for you guys, and whoever preparing for NEET, right, within our school schedule, it's up to you. Whenever you find free, you can access those videos at any point of time. Okay, but especially the full chapter videos you need to go through, especially the 11th botany also I'm going to cover. That is a main prime motto behind taking this taking live session at 7 to 8 p.m. Using projector and videos to enhance your understanding on the plant physiology and also animal physiology, the 11th portions. Okay, so don't miss it. Okay, this is an announcement I wanted to make for you guys. So whoever find it you know that useful, you can share among your friends, metric friends as well, so that everyone would get benefited, right? So that's it. Yeah, 7 members. What about others? Okay, we'll start then. We already waited a long time. 5 minutes already over. So yesterday we were talking about the lesson principles of inheritance and variation. Right, whoever not attended the last session, the last class video is already posted, naming principles of inheritance and variation outline, right? Chapter 5 NCERT will be there. So you can go through that and then come back to this video because this is continuation video. So in this, yesterday we talked till the linkage and recombination and sex determination also we talked about. So in the NEET perspective, right? In the NEET perspective, how to handle these topics we discussed yesterday. So let's move forward in this lesson. That is, next topic would be good morning, Kritika. And then moving to the next topic that we're gonna discuss is mutation and genetic disorders. I don't know this lesson, the disorders and their condition and their symptoms and the genotypes are very important. So mutation concept you already knew. We have studied mutation concept in two to three different separate chapters. But when we talk about mutation at the molecular basis level, or here what we talk is point mutations. When point mutation occur, even a molecular basis of inheritance, we covered that point mutation leads to sickle cell anemia, and we know which amino acid change, etc. Right? And then frame shift mutation, and then chromosomal aberrations. Here the terminology you should be following in class are already mentioned: aberrations, abnormalities, anomalies. All these three are anomalies. All these three are synonyms, right? So during this, because of this chromosomal aberrations, right? Mostly you will see these chromosomal aberrations in cancer cells. Right. At the same time, we will be talking about the mutation causing agents or mutagens, which we covered in human health and disease. Once quickly go through. So physical mutagens and chemical mutagens, which causes mutation. So this is a mutation concept within this lesson. So you will be getting like frame shift and point mutations, examples of the diseases, etc. So that is a aspect, right? And here we already, in running notes, we already mentioned very clearly that, loss of chromosome and gain of chromosome. We talked about euploidy, aneuploidy, right? And then how many chromosomes, which chromosomes, for example, whether they are autosomes or allosomes, like sex chromosomes, Klinefelter syndrome, if you take one of the extra copy of the sex chromosome. Likewise, you need to focus on the genotype, which chromosome is showing this abnormality and what is its number, and disease symptoms as well. So you may get statements, correct or incorrect statements when it comes to disease statements, disease this disorders, symptoms. Okay. So moving forward, so we'll be talking about here is pedigree analysis. In academics. So we done a lot of pedigree, right? Once go through your running notes and see that each disease has got its different what do you say, parents. We took different parents, like affected male with the normal female, affected male with the carrier female, affected male with affected female, like that. We took several combinations and we did different process, right? That's how we did this genetic disorders. When we talk about this pedigree analysis, once quickly go through your what do you say, running notes that I have gave already, both NEET and academic students. Okay, so pedigree analysis, much you don't, no need to touch much on the theoretical aspects, like what are the symbols, female, and then, but what you need to focus is that diseases. So when we talk about diseases, right, they may give the pedigree and they may ask you to find out the genotypes. So this is what you need to focus. So the genotypes you need to you have to be able to chart out the genotypes. So I already gave certain rules to follow when we do this pedigree analysis, right? Autosomal dominant, autosomal recessive, sex-linked dominant, sex-linked recessive, and the criteria to meet those pedigree. How to start the pedigree analysis, I already gave enough idea in our academic session, but all these things again I will be covering in brief outline session, that is 2 to 3 p.m. whenever this topic comes. I will notify you, right? So here, let's move to the diseases here. Nothing but the genetic disorders can be of Mendelian disorders or chromosomal disorders. So Mendelian disorders are something which is change in the DNA sequence or single gene, right? Mutation, alteration, whatsoever, that there would be phenotypic expression would be changed. So that leads to disease. It means the pattern. These disorders shows the Mendelian pattern, right? The pattern of inheritance of these Mendelian disorders can be, can trace by the pedigree analysis. So if you take this hemophilia, color blindness, and sickle cell anemia, phenylketonuria, right, and thalassemia. Thalassemia, we already discussed about two types of thalassemia. At the same time, cystic fibrosis. So Mendelian disorders and right, and their examples are important. So if you take this, first we talk about, we'll take autosomal dominant and autosomal recessive. I already mentioned in during our discussions that how to approach a pedigree chart, right? Sometimes they give information, but most of the cases they may not give the information that whether it is a dominant trait or recessive trait, that is parents. They may ask you to find out even traits, right? Nothing but this genotypes of the parents, or within the F2 generation, you have to find out, in the second generation children. Second generation, you may be asked to find out the deceased children as well. So you have to get the genotypes correct. Means you should have an approach to this pedigree analysis, which we already discussed in academic classes, NEET classes. So make use of that running notes, right? And then if you take this disease by disease, hemophilia, which is otherwise called as royal disease, right? Because we have seen this, identified this hemophilia in a royal family, and there is a huge loss over there, and there is a carrier female which actually transmitted this inherited this disease, that is hemophilia. So in this, genotypes are important. It means which genotype actually male would become hemophilic. Normal male, what is the genotype? So those are the genotypes and everything which I already mentioned in class, as well as I'm giving in outline, today's outline. You can find it also. So which genes and which alleles that actually causing the change? It means, for example, there is a non-stop bleeding will be there. So the traits or the characters or the genes which actually control the blood clotting are getting affected in this hemophilia when they have this genes, right? So likewise, what is the condition of the female, whether they are hemophilic or carriers? If they have to be hemophilic, what should be the condition? So these are the aspects you need to focus in each of the diseases. So next, when we talk about the color blindness, the same way the genotypes are important, and which would be the carrier. If the carrier female is made coupled with the married to a normal male, what is a generation 2? I mean, the kids would get what sort of genotypes, like that combinations you should practice when you do this genetics, especially this pedigree analysis. Different combinations we already did in this. So when we have a carrier, but you have to remember that in sex-linked you will not find any carrier in male, only female you will find the carrier. That's a thumb rule. And then coming to sickle cell anemia, and the genes, those are affected, right, which are homozygous dominant, HPA, capital A, H, heterozygous dominant, and homozygous recessive, HPS. So these genotypes are important, right? And which amino acid is changed to which one during this mutation, this single cell point mutation, sickle cell anemia, which led to the normal RBC to become a sickle cell. Both it has advantages and disadvantages as well. So which chain of hemoglobin got affected? Beta globin chain of hemoglobin HB would be affected, and from which amino acid to which amino acid they change, and its codon also important. Is that clear? So if you say this, This sickle cell anemia is caused by substitution of glutamic acid by valine. So valine is replacing the glutamic acid at the 6th position of beta globin. So this is utmost important, because they may give 6th position, 16th position, 60th position. So you will be remember only 6, but you may get confused. So you should be very much clear with the which amino acid replace which one, and its codon as well, right? So that is the important aspect. So for example, GAG to GUG. So GAG to GUG is what the substitution occurred, right? And what leads to this? So if the sickle cell anemia comes, what are the symptoms you see, right? So RBC biconcave disc structure would be converted into sickle cell, etc. So next, coming to the phenylketonuria. Phenylketonuria is one of the metabolic disorder, and it should be very much carefully understood, at the same time remembering, right? And this comes under autosomal recessive disease, phenylketonuria, and in this enzymes are important. Enzymes are important. So which enzyme actually responsible for conversion of phenylalanine to tyrosine? But in the absence of this phenylketonuria, right? In the absence of this phenylalanine hydroxylase, right? This enzyme is there, right? So in this phenylketonuria, in the absence of this phenylalanine hydroxylase, this phenylalanine is not anymore converted into tyrosine. The accumulation of phenylalanine would be seen in the body of the system of the this deceased person, right? So that then how what happens as a result? The phenylalanine will be accumulated and converts into phenylpyruvic acid, right, and other derivatives. So you should be focusing on the enzyme here. At the same time, if this enzyme, nothing but phenylalanine hydroxylase, right? Phenylalanine hydroxylase, if this is deficient, right? What happens? So there is, we know that this all in biomolecules, we already learned that these all reactions, chemical reactions would occur in the presence of enzymes in living systems, most of the reactions. And when this enzyme is not there, there won't be any reaction. Enzyme substrate complex and then enzyme product, right? So but there is no enzyme, so what happens is the substrate, nothing but phenylalanine, would stay in the system itself, and it eventually converts into phenylpyruvic acid and other derivatives. So this accumulation of this these products and derivatives, phenylpyruvic acid and their other derivatives, accumulates in brain, resulting in mental retardation. So this is the aspect you need to focus on. So in this lesson, in this today's topic, especially these diseases are utmost important, and you may get confused. So take time, spend at least half an hour per disease, so that you have some space to study. Like the thalassemia, we have two types: alpha thalassemia, beta thalassemia, and again. So are you able to follow, guys? Are you able to follow, guys? Yeah, Shamali. Yeah. So let's move further. That is okay. Okay, Daksha. Fine. So in thalassemia, we have two types of thalassemia, and you should not get confused with alpha thalassemia with the beta thalassemia, right? And then chromosome numbers, right? In alpha thalassemia, what you see is the this alpha globin chain. That's what it called alpha thalassemia, right? The alpha globin chain is affected, right? So this alpha globin production, the same protein production, is controlled by a gene, right? Which are linked genes. That is HBA1 and HBA2 on chromosome 16. For example, you guys 16 years old, right? So you remember 16, right? So when you're studying 11th standard, you are 16 years old. So first one, alpha, right? alpha chromosome 16, and the beta in beta thalassemia, beta globin chain will get affected, and it is controlled by single gene, that is HBB, which is present on chromosome number 11. Is that clear? So that you need to take care. So they may give this hemoglobin genes, right? I mean, for example, HbA1, HbA2, HbB, and then sickle cell anemia. The change in the gene sequence on chromosome. So these are the three different types of gene sequences, and they may give chromosome numbers. So you have to or the diseases they may give. So you need to identify which chromosome number that this gene is affected in which disease. So this is what little confusing thing you may observe in this thalassemia and sickle cell anemia. One is qualitative and one is quantitative. Thalassemia is something which is quantitative disorder. It means the production of hemoglobin, right? The production of this globin molecules would be reduced. But in the case of sickle cell anemia, it is a qualitative problem. It means that the hemoglobin, this globin molecule itself is incorrect functioning will be there in the sickle cell anemic disorder. So that's what the difference between thalassemia and sickle cell anemia. So in the thalassemia, alpha and beta and their chromosome and their genes, whether they are linked or whether there are two genes controlling one phenotype or one gene is controlling one phenotype, is what you need to focus in this thalassemia, right? So and then comes to the chromosomal abnormalities. So we have talked about during class, right? During NEET class and academic classes, both the classes. We talked about aneuploidy, polyploidy. Aneuploidy is something which is gain or loss of chromosomes due to failure of segregation. So in cell division, we already studied about the segregation. Yesterday we talked about the segregation of chromosomes and the alleles which are present on the chromosome. These two occur in pairs. Alleles occurs in pairs and they separate individually during gamete formation. And even chromosome occurs in pairs, homologous chromosomes, and they segregate individually, independently, they assort during the meiosis 2. That is, at the end of the meiosis, what happens is this chromosomes reduce to half, and then we get, this individual chromosomes go into the, one set of the chromosomes will go into one gamete. So during this process, what happens is there would be sometimes a disjunction occurs, like they don't separate or they don't segregate. That leads to either gain, because if one chromosome is present extra in one cell, means the other cell lost it, right? Either in that way, the one cell exhibit gain or one cell would exhibit loss of chromosome. So this condition we call it as aneuploidy, and we have a euploidy condition, that is, or euploidy or polyploidy, where it is an increasing whole set of chromosomes. During in academic classes and NEET classes, I gave all different conditions. So polyploidy, what are the whole set, which whole sets, and what is the disease you see, right? So failure of the cytokinesis after telophase stage of the cell division leads to this, euploidy or polyploidy. So that the increase in whole set of chromosomes which you see in polyploidy, that's what polyploidy. Okay, ploidy is something which we talk about the chromosome number, right? So when it comes to the examples of chromosomal disorders, right, we have got Down syndrome, Klinefelter syndrome, Turner syndrome, right? So these are the three important disorders that we cover in this chromosomal abnormalities, aberrations. We cover anomalies. You guys are following? Right. So I told, right, anomalies or disorders or aberrations, chromosomal aberrations, all these are synonyms. And if you see this, in this we cover Down syndrome, Klinefelter syndrome, and Turner syndrome. Here you need to remember that which chromosome is extra, which chromosome it lost in case of its any loss is there, right? Whether they are autosomes or allosomes. So this representation of genetic composition is very much important, along with the features of this disease. For example, they may give the features correct or incorrect: broad flat face, furrowed big tongue, and partially open tongue, open mouth. So that is feature of Down syndrome. The person affected with the Down syndrome, and it exhibits trisomy of 21st chromosome. It's an autosome, right? So one additional copy of 21st chromosome would be seen. So you need to figure out, or you need to identify these options in the given question, right? That which is trisomy, which is in which allosomes one of the sex chromosome, X chromosome, is extra. In the case of Klinefelter syndrome, right? Gynecomastia. If you see this, gynecomastia is something which is what do you say, the feminine characteristics or development, bodily development would be seen in this Klinefelter syndrome affected person, where there would be an additional copy of X chromosome. So it is 44 autosomes plus XXY. So one extra copy of X chromosome will be seen, and so total 47 chromosomes, right? So it is something addition. And then comes to the last one, that is Turner syndrome. So Turner syndrome turns to be, the loss of one chromosome. The one is nothing but the Y chromosome. If you see this absence of one of the X chromosome, sorry, it is one of the absence of X chromosome, so that the feminine characteristics would be not formed. It means the X chromosome controls the secondary sexual development, etc. Right? It determines the sex. So what you see is the futures is the sterile ovaries, or sterile or ovaries would be rudimentary, right? They are not useful. So lack of other sexual characters also will be resultant in this Turner syndrome, because of why? Because X chromosome, one of the X chromosome is missing. Chromosome will be there. So it is 44 autosomes plus 1 X will be there. So total 45 chromosomes. So this is about this lesson, right? So any doubts in this? I hope this quick go through of NCERT would be useful for you. But the outline, the brief outline, will be given from today. From lesson to lesson, I will be posting. So nothing to worry about. So just keep following what I'm posting, and I will be sharing the links as well. Right? Okay, let's move to the next lesson, that is environmental issues. Excuse me. So let's focus on the next one, that is environmental issues. Right. Now for today, material and yesterday's material, this study material, worksheets and all, I arranged and even test question papers also arranged in website, right? So that link I'm sharing. So today's also you can download from here, and I will be posting in group as well. No problem, but you can directly access from this also. Okay. So this is a link. Fine. So let's move to the next lesson, that is environmental issues. So if we talk about environmental issues, right? So this lesson is one of the lesson which which covers a whole lot of issues that are occurring within the environment, how this affecting the entire global biodiversity, etc. that we will try to cover, right? So how does this environmental factor would really affect So in this environmental issues, will be more focusing on the pollution. So pollution is something which is undesirable change, right? Undesirable change in physical, chemical, or biological characteristics of air, land, water, or soil. So we have to focus on these four aspects, that is air, land, water and soil. We will be focusing on this, and then we will be talking about the pollution and its permissible levels and its control and what are the pollutants actually we see, right? Many people would post in social media saying that now the earth is healing, the ozone is healing, and etc., right? And that too they post in the social media using the cell phones. So I don't get one thing, that when the humanity is concerned, right? When we wanted to really enjoy all this setup, now see we are able to interact. Even there was no physical regular conventional classroom setup, we are able to meet our criteria, that is NEET preparation. So that's how we developed, right? So we are not caveman to sit in a cave and we can't say hoo, like. So we develop, right? So when the development is there, we can't, development and environment will not go hand in hand. So every time there would be imbalance will be there. It means when you focus on the development, right? Environmental degradation is obvious. As a human, we are the just part of the entire this universe. We can't control, or we can't really save the earth, right? But but at certain aspects we can actually control the aspects of the pollution, unwanted pollution, right? For example, now people are saying that the earth is healing, and the this ozone layer got at the Arctic Circle we have a ozone hole is there, that is actually the ozone layer depleted, and then now it is healed. But once again this lockdown is removed, what happens? Everyone would be using cars, everything would be normal. So what we can restore is actually nature would restore itself. We human part of this entire universe, we can't really take over the nature's plan, right? So but that's what people would keep posting, that no, save earth. Who we are to save earth? That's the thing, like how we gonna save. But here we need to remember that we can control the pollution, but we can't really completely eradicate, because we are connected with the development, right? We humans tend to develop. That's what our brains do work. When we talk about Homo habilis and now Homo sapiens sapiens, that's a difference, right? So we started thinking, and we do this entire developmental process. And when development increases, we can't really protect the environment. But I'm not suggesting to destroy the environment intentionally, but we don't do intentional damage. If people does an intentional damage, we have to protect and we have to control. So in this lesson, that's why I put this lesson even first. That's environmental issues. This is the perspective we need to have, realistic perspective, right? So when we talk about air pollution, and we are right from the childhood we are studying the air pollution, etc., right? So what causes the air pollution, harmful effects of the air pollution, and how to control. So these are all correct or incorrect statements you may get, right? So yeah, so especially you need to know the conversions. So unburnt, right, half burnt or unburnt hydrocarbons, right? How do they actually convert, right? How can we convert, or what are the actual pollutants in the air, right? So motor vehicles, motor vehicles, through motor vehicles or electrical appliances, home appliances, we tend to release a few pollutants as well, right? Which actually have to have a permissible limit as well. So this causes harmful effects of the air pollution, is something which reduces the growth and yield of the crops, and even premature death of the plants also maybe seen. At the same time, humans respiratory problems, for animals respiratory problems and irritation, inflammation, right? The allergic reactions also may develop, or even premature death. If you see the entire India, right? So we have an Delhi. If you see the Delhi, right, North India, Delhi. If you take that, it's highly polluted. So metropolitan cities, if you consider any metropolitan cities, would be highly polluted, right? Gobi is little exception actually. So likewise, in all this air pollution, what you need to focus is what are the harmful effects and how do we really, control them. For example, catalytic converters. Motor vehicles should have the catalytic converters to really convert this, this harmful gases, right? Or electrostatic precipitators, right? The particulate matter which is released into the air can be precipitated electrostatically. And then comes to the noise pollution. The noise pollution, we no need to discuss much as well, right? There is an act, that is Prevention and Control of Pollution Act, that is Air Act, that is 1981, right? Noise is also at the undesirable high level of sound. Nothing but noise is nothing but undesirable high level of sound. So morning when I wake up, right, even before opening eyes, I can hear a lot of noises from different areas. I don't need to mention from where, but that's how the noise begins. Our day begins in India, right? We don't really care about the noise, right? But at the undesirable levels we call it as noise, and it has an effects, right? So here you need to focus on how much decibels, right? If you take the sound level above 150 decibels, right, maybe damaging the eardrums. So you need to focus on these numbers. So how do we control the noise pollution? Right, we have to delimit the usage of the loudspeakers, and then during especially the cultural aspects of India or any other country, that we fire crackers, for different occasions, that needs to be reduced, right? So that it can really control both noise as well as air pollution. And then laws and policies in India needs to be a little should be forming little stricter. For example, if you take this vehicle pollution laws, right, we have got you have to focus on what are the laws present in India to control these aspects, air pollution, right? Are you able to follow, guys? So because it's 12th standard, maybe portions, maybe very few members came, but please do remember that when we start the other lessons, you have to be everyone should be coming. How do you guys clear? So in this lesson, what you have to focus is what are the pollutants, from where it is released, and what are its effects, and how do we really control this pollution. So these are the three. This is a basic skeleton for this lesson. So all this thing you can make a tabular form. This is noise pollution, this is air pollution, sound pollution, noise pollution, water pollution, and pollutants and its effects and its controlling measures. And based upon this pollution, type of pollution, what are the acts, laws are present in the in India that you need to focus. Okay, you guys are there actually. Please do respond. Is there any problem that you guys are not responding at all? Yeah, you guys are there. Yeah, please do respond, guys. So that is it for air pollution. And when it comes to water pollution, we already talked about in microbes, in human welfare, we talked about the sewage treatment, at the same time BOD levels, right? Biochemical oxygen demand, and how do this affect the quality of the water, right? So how can we really check the quality of water based on the BOD or using this BOD, right? So here again you need to focus on the different pollutants that you see in the water. For example, what are the biological, this algal bloom formation, right? So in the presence of more nutrients in the water encourages the growth or excess growth of this planktonic algae, which causes algal blooms, right? Which actually affects the organisms which are underneath them. So at the same time, they are extremely toxic to human beings and animals as well. So Icornia, if you see the Icornia, terror of Bengal. So this is something which is unique way, unique example. So you need to focus on this example, that is terror of Bengal, which is a weed, right? Aquatic weed, which actually uh, it is spread abundantly or grown abundantly in eutrophic water bodies in the West Bengal. So that we got it as a terror of Bengal, right? So at the same time, industrial waste like DDT, heavy metals like mercury, cadmium, copper, lead, etc., and other organic compounds may actually, you know, makes. So, sir, why it is known as terror of Bengal? It's network problem. Okay. No, it's a kind of a network problem. Just let me check once. Yeah, is it okay now? So if you see this, yeah, someone asked Chetan Kingdom. They asked one question, that is why it is known as Terra Bengal, because this Icornia is there, right? So this Icornia, grown in an exponential, exponential. It means that the rapid growth and alarming rate, right, which spreads on the surface of the water body completely. It just spread with this Icornia, right? This water hyacinth. So this Iconia spread the entire water body. It means what happens is the underneath organisms would really suffocate. They don't get enough nutrients. At the same time, they don't get enough light, and they eventually they die. Right. At the same time, this cut out the light, and it also causes the oxygen increase in oxygen demand, so which causes the death of the fish and other aquatic organisms. So that's what nickname, or the other terms we call it as terror of Bengal. It means it just polluted this entire ecosystem. So that's why we call it as terror of Bengal, where we observe this, because it even competes with the other local aquatic organism. That's the main aspect. Is that clear, Chetan? Right, let's moving further. Right, here we talk about the sedimentation. When we talk about wastewater management, we already covered in this microbes in human welfare, right? How do we do that? Sedimentation, filtration, chlorine treatments, and other. We have this entire primary treatment, secondary treatment, biological treatment, etc. So which actually lowers the pollution. So you need to focus on which waste we are talking about here. So integrated wastewater management, if you take, so we are talking about industrial waste, right? Biological waste, how that needs to be treated, what is the source, what are the effects, and then we have to talk about the biological magnification, right? This accumulation of toxicants like mercury and DDT, and then it eventually enters into the food chain. So if these toxins are actually released into the the lake, the ponds and lakes, right? So the fish would feed on it, the fish we will feed, and that's how the toxins would enter into the human system. This process we call it as biomagnification. And this biomagnification flow chart is important. I gave it in material as well, right? Outline. So you please go through that, right? So here water contaminants like nitrates and phosphates. Now how do they affect the water? You just focus once. So if you see then how to really how to address this issue, control this issue, is that eco-san, that is ecological sanitation, is actually taken up by Kerala and Sri Lanka. That it is something, a sustainable system for handling human excreta using dry composting toilets. So that's how we are actually handling. Now we are talking about controlling aspects. Water, what do you say? This water pollution and the pollutants. So far, what we talked about different, we talked about biological magnification. We talked about industrial wastes, right, and then eutrophication. The what do you say? This algal bloom formation, right? And then how do we control ecosan, right? So ecosan, which actually even one of the controlling measures to actually control the pollution in the water bodies. So what are the other laws that are present in the controlling water pollution? You need to focus, right? That is Water Act 1974. So you may get this Water Act, Air Act, right? In one column A, and the column B will be giving the years as well. So you need to match the following. So you should be very careful about this. So wastes and their when we talk about waste, the other wastes, right? So when we can classify these wastes into different types, like solid wastes, right, which are both biodegradable, non-biodegradable, and recyclable. For example, hospital waste, e-waste, like this entire systems and electronic wastes, right? So all these wastes, how do we know their examples, or where you need to focus on, right? This biohazardous hospital waste, how to handle, right? Otherwise, now COVID-19 situation, this PPPs, right? This personal equipment, right? So what we have is these are not handled properly. They were thrown thrown on the streets as well, so which would be contaminating the other humans or animals. So you should be carefully discarding this gloves, hand gloves, and other masks, etc. So likewise, different types of waste you will be having, and how to handle, you need to once focus. And then coming to the next aspect, that is when we talk about organic farming in strategies for enhancement of food, we already talked about how do we shifted, for example, from using this artificial fertilizers to organic farming. Why now every farmer is actually encouraged to go with the organic farming, because the environmental effects and the effects of fertilizers, chemical fertilizers, is more harmful. And then even this organic farming produces zero waste, right? So this zero waste procedure is what actually one of the integrated organic farming techniques, right? uh, so these are the aspects which we can control. This one of the solid waste or chemical fertilizers, right, by adopting organic farming. So this is one of the what do you say one of the controlling measure. So here we have Haryana Kisan Welfare Club, right? So what is this welfare club talks about? And it's actually spreads information regarding the practice of integrated organic farming. So this name is important, because this is conducted by NTA mostly. This NTA, this North Indian, what do you say, this the names which we talk about, Kisan, these words are very important when it comes to the preparation for any competitive exams, right? So especially when they first started, and now when they're given in the textbook, you should not leave those behind. For example, Haryana Kisan Welfare Club. So they talk about, or they spread information about the practice of integrated organic farming. As I already told, this is a very quick highlights and outline of the chapter. I'm not explaining much on any topic. Okay, but the other videos I'm going to release, I will be explaining much, in depth. Here because of the time constraint, we are at the same time for NEET preparation. Already these lessons were taken, so this is a better approach, right? Okay. So the next aspect is the greenhouse effect and global warming. It's a very old topic at the same time. Here you need to remember the greenhouse gases and their contribution in the global warming. So if you take carbon dioxide is 60%, right, CFC is 14%, N2O it is 6%. So this composition and percentages are very important, right? And then especially the greenhouse gases and its effects, right? And then next we will be talking about the ozone depletion in stratosphere. So when we talk about ozone depletion in stratosphere, just now I mentioned also, when we talk about development and the environment, they both can't go hand in hand, right? So when always human sees for development, we can't really stop the development and sit in the homes forever. So we have to find out some controlling measures and intentional damage to environment. Okay, if something which is unintentional, that is a different thing, where the nature, anyhow the nature will fix, because earth is not just 10 years or 20 years old, okay, planet, right? It's a few million years before it formed, and it knows how to really restore the conditions, environmental conditions. It doesn't see that whether you're human or bacteria. It doesn't have that sort of consciousness, but it has its own sort of restoring restoring itself. Okay, but we have to be adapting to the restoration, as simple as that. If you are unable to adapt to the earth changes, for example, it's like huge temperature increase, huge temperature drop, right? If you are unable to adapt, just like dinosaurs, humans also will get extinct. That's it. But we can't really control 100% of nature. Okay, because we are part of nature. That you need to remember. So when it comes to ozone layer depletion, what causes ozone layer depletion, and the Montreal Protocol. So here, if you see here, what are the protocols that actually introduced to safeguard these ozone layers? That's what you need to focus. Right. Finally, if you see this degradation of improper resource utilization and maintenance, for example, intentional damage to environment like deforestation, right? That actually countered by this chipko movement. So this chipko movement, these important movements are important when it comes to this defo deforestation or other aspects which are covering in this improper resource utilization and maintenance, right? So in this degradation of environment by improper resource utilization and maintenance, which actually focus on deforestation and its counter movements like chipko movement, that you need to follow, right? Okay, so these are the aspects that you see in environmental issues. So here you need to more focus on the conservation acts and the other laws that are given in the by Indian government, for that matter international governments, UN, whatsoever. So those laws you need to focus, right? At the same time, what are the management styles we can develop? These things would may come in correct or incorrect statements, right? So with this, we completed this lesson, that is environmental issues. So I just quickly went through the important aspects. I didn't go through the entire chapter line by line, but because we are taking two lessons at one shot. So I hope this would be just brushing up your preform knowledge as well, right? So that is it for today. And as I already told that uh, this brief outlines would be released from today, and even full chapter videos will be released today. And kindly subscribe and like the video at the same time, and share to your metric students and metric friends and other friends as well, so that everyone would get benefited, right? So any doubts you can ask. So these are the two lessons. That link is given, right? That link you will find the tests, and as well as you will find the material for today as well. So you can prepare at home. And if you have any doubts, you can ask me at any point of time. I hope this session is useful for you. So if anything, any doubts out there, please ask. Okay, I'm ending the session.